All right, let's kick this off, shall we? Hello, everyone. Welcome. Good evening or good morning from wherever you're calling in. My name's Pablo. I'm a Reef Check ambassador, and I will also be your host for this evening. And welcome to July's annual Coastal Coral monthly talk, online edition. So I do just want to begin by introducing Gail. Tonight's talk will be on an introductory guide into coral taxonomy and everything coral related, and I am super excited about it. I do also want to acknowledge the traditional owners of this country. They're the Kabi Kabi people where I am currently situated in the Sunshine Coast. But I do want to acknowledge all of Australians, Indigenous peoples, past, present, and future elders. Yeah. So before we get into everything, we just have a few little details. And one is, who is Reef Check Australia? So for anybody, if this is your first time coming into one of these talks, Reef Check Australia is a non-profit organisation mainly based in southeast Queensland, here on the Sunshine Coast where I am, and Brisbane. We empower local citizens like myself to come and volunteer and preach about citizen science that we do. We do hands-on research by going out and surveying local coral reefs each year to get some big data sets and monitor the long-term health of our marine ecosystems, as well as hosting local community events and engaging our community to try and educate everybody in our local area about what's up underwater, what's going on, and how they can minimize the impact on our marine life by changing a couple things in their lifestyle. Just a couple housekeeping etiquette if you aren't already acquainted with Zoom through the past year or two, however long it's been so far. If you could please mute your video and your microphone throughout the talk, just so we don't get any overlaying or unwanted audio coming through. And it just helps with the bandwidth as well to keep everything going nice and smoothly so we don't lose Gail halfway through her engaging speech. If you do have any questions today, Gail has also asked, excuse me, that you just pop it in the chat, ask your question, and I'll kindly butt in and unmute my mic and ask it for you if you like. And just as we finish up, before I hand over to Gail as well, we will take a quick group photo just to post on all the socials to get it out there, what we're doing. And if you aren't on the mailing list, we do have a monthly mailing list with all our local events, heaps of cool information, as well as a couple extra things as well. Please email that email there, seqevents@Reef Check Australia, and you'll be added. Otherwise, head to our website, Reef Check Australia. I do want to give a couple shout outs to a few events going on in our local community as well. Currently, we have Plastic Free July going on, which is always a giant hurdle for me, I know personally, to overcome. It's a global movement that just tries to encourage people to be more conscious about their plastic consumption. The aim is to try and go an entire month without using any plastic. But if any of you are trying this right now, it is virtually impossible unless you change your entire lifestyle, really, and be extremely proactive. But in my personal opinion, the aim is not to be completely plastic free. It's to dramatically reduce your plastic, or at least try to reduce it as much as possible, really. And you kind of have to do it in small steps. Like the first step I did was buying a couple cute tote bags and some reusable coffee cups, trying to have a better waste management system at home, doing some composting, reusing or just not using plastic bags where necessary, and being more conscious, really. I don't eat Chinese takeaway food anymore, which is really annoying because it's delicious food, but dining in and taking other things. But there is a website with heaps and heaps of information, plasticfreejuly.org, about anything else if you want to educate yourself on that. We do also have, if you're in the Sunshine Coast region, an upcoming event this weekend, I believe, Saturday at 8 to 12. It's Spring into Scavenge, it's called, at Caloundra Park. It's a cleanup event with a bit of a twist on it. It's very family friendly, but they go and clean up the beach and turn it into a scavenger hunt, really, and also engage the community and show them the process of cataloguing plastic pollution that washes up on our shores. So it sounds like a lot of fun. So head on down if you're free. Yeah. We do also have another festival. It's called Peaks to Points. There is a family day this weekend as well, Sunday the 18th July. However, it does run for the entire Plastic Free July month, actually. It's based in Brisbane, and the event this weekend is at Oxley Creek in Rockella. And if you Google Peaks to Points Festival, they have all the other events on their website as well. It's a really cool website. And then we have another almost month-long festival due to COVID, which is Horizon Festival. This one's based in the Sunshine Coast. And this isn't entirely an environmental festival, but more of an environmental / social festival. So it encompasses a lot of cultural events. It sounds like loads of fun. Reef Check Australia will be actually at one of these events. I'll be there personally, so you can see my beautiful face up close and personal at the University of the Sunshine Coast. So head on over to their website, have a browse at all of their amazing events that will be going on, and choose which ones you like. And we do have another amazing Coast to Corals talk next month. We are lucky enough to be blessed with a collection manager from the Queensland Museum. So it's going to be super fascinating. It's about carnivorous sponges and other sessile marine invertebrates. And it's going to be given by Dr. Merrick Atkins. So that will be next month on the 10th of August. Registration will be opening soon, but just be on the lookout for another Facebook event page that will be popping up in the next week. And if you also are on our email list, you can't miss it. We'll be sending it out to everyone as well. If you do want to get involved, become a volunteer and a friend of mine, and the answer is yes, who doesn't, head on over to our website again. We do two types of volunteering at Reef Check, which I really love. We do ambassador volunteering, where you're a educator like myself and you engage the local community, as well as survey diver. So if you're a scuba diver and if you're passionate about helping out our local reefs, you can also become an Australian qualified survey diver, where you go out and monitor our local reefs. It's free diving, PS, by the way. So really, it's a win-win situation. Yeah, head on over to our website. And in case you aren't already, but I'm sure all you lovely humans all are, please follow all our pages. We're most active on Facebook and Instagram, where we keep you all up to date with our events that are going on in the local community, any information, politics, anything current and relevant, we post it on Facebook. So we're at Reef Check Australia on Facebook, the same on Instagram as well. We are on LinkedIn and Twitter. We're not as active, but we are there as well. But Facebook's a great place. We have Facebook events with all of our relevant info to help you guys out. We do have a couple thank yous before we get started. A huge thank you to all these local and non-local businesses: Sunshine Coast Council, Mask Events, Townsville Council, the Port of Brisbane, the City of Gold Coast, and the Clean Water Group as well. Events like these tonight even wouldn't be possible without the support, financial and non-financial, of our local community, really, like you guys, and with businesses like these. And it's not an updated photo, but I do want to say a big shout out and thank you to all the volunteers that helped make this happen. Particularly on this call, there's Erland and Lindsay, who are both ambassadors as well, who help facilitate these events. These are, Reef Check, as I said earlier, is a non-profit organization, which means that we don't take any money. We do all of this for fun and because it's our passion, and it wouldn't be possible to have these awesome nights without you guys. So thank you very much. And lastly, but not leastly, a warm welcome to Gail Riches, tonight's speaker. So a little bit of background info on Gail. She's a scientist, a scuba diver instructor, and a marine educator. In 2018, she created a website dedicated to helping marine science teachers and students across Queensland in implementing better and easier education plan for our local schools to promote the stewardship and our local oceans, really, through collaboration. She has a Bachelor of Marine Science and Management and a graduate diploma of secondary education. She works as a teacher at St. Teresa's Catholic College up in Noosa, and as on her business, the marine science as well. Prior to teaching school, she worked as an education officer at Sea Life down in Mooloolaba, and as a dive instructor as well for PADI in heaps of places across Queensland, as well as the Mediterranean Sea. So without further ado, I'll hand it over to you, Gail.
Thank you. That was a lovely introduction. Thank you, Pablo. Hello, everybody. Thank you for coming. I will share my screen. I have a PowerPoint to share with you.
I always forget. I do want to ask a couple poll questions before we begin as well. Sorry, yes. I jumped the gun.
Okay, I'll stop sharing. Hang on.
And then we will also, if everyone wants to quickly turn on their cameras, we'll take a group selfie as well. So we just use these polls as kind of analytics to gauge where our audience is from, so we can kind of target our market a little more to get as many people to these events as well. But thank you all, 40 of you, for turning up tonight. It's a great audience. And after we end the polling in a couple more seconds, we'll get a nice group photo. Welcome to everybody. It's so lovely to put faces instead of just seeing black boxes everywhere, huh? Awesome. Awesome, awesome. And I do want to remind everyone again, this is being recorded, this lecture. So if you don't want your face or your name being on any of our social media sites, even though they're all beautiful, just turn your camera off and change your name on the right there. But it won't be too much of a problem. All right, now everybody smile. And I'll get some print screens. Awesome. Let me see if I took that correctly. And then we can move on. Awesome. Thank you so much, everybody. Sorry to interrupt you, Gail.
Okay,
if you want to get to it now.
It's good to see everybody's faces too.
Yeah, it is. It is. You actually have an audience. They're not just robots tonight. So I'll let you get to it now.
Okay. Can you see that? If I share.
Yes, we can. Yep, that's perfect.
I'm just gonna put that there. Okay. Yes, so thank you for coming along. It's lovely to be here. And so this talk will be an introduction to coral taxonomy. But first of First of all, I'd like to thank Coral Veron for giving me permission to use a lot of his images that he has on his website, which looks a little bit like this. And I've circled down the bottom species fact sheets, which has some fantastic information and some unbelievable photographs, of which I've used a lot of here. The authors there you can see on the right-hand side, Charlie's up the top, Mary, Emre and Lyndon. So thank you to them. They also have that information as a 3-volume book set, which is a fantastic resource on the left there. On the right is an introductory guide to coral taxonomy that I wrote for high school students, which is very similar to what I'll be talking to tonight. And you can find that on my website, marineeducation.com.au. So plug in. Some other fantastic resources if you're interested in going into this into a bit more detail. Coral Finder 2021 is Russell Keeley's work, and one of the best underwater coral ID guides, I think, that there is, and it's really, really simple to use. And it's under bio guides. It's recently been updated, so that's a fantastic resource as well, and it's really affordable, and there's some fantastic information there. On the right is another great coral ID guide from Western Australian Museum that is available online. There's some fantastic videos that you can watch provided by James Cook University and Dr. Mia Hugenboom. And then I have my website as well. So I'll throw that in there. So with no further ado, let's begin. Hopefully by the end of this talk, you will be able to identify these hard corals here right down to genus level, based on key characteristics that we use to identify the genus of hard corals. And so hopefully I will show you this picture at the end of the presentation, and hopefully you'll be able to identify some of those things that we're going to be talking about tonight. And if you remember the name of the genus, that's even better. Okay, so all of those are hard corals. So we're just focusing on hard corals tonight. It is just an introduction. There are lots and lots of hard corals. So we're just going to really focus on the ones that you see quite often on the reef and are easy to identify. Now, hard corals, another word for hard coral is scleractinia, and that comes from the order Scleractinia. This is how they are classified in taxonomy, where they're given a genus and species name. And so if we start at the bottom of this slide for kingdom, they are animals. They're not a plant. However, there are some plant representatives living in the corals, which you're probably already familiar with. And that is an algae called zooxanthellae. And the zooxanthellae live as a mutual symbiotic relationship with the coral. It has a nice place to live, and in exchange, it carries out photosynthesis and creates lots of oxygen, which helps the coral, and it feeds the coral as well. So not all corals have this relationship with the zooxanthellae, but certainly the ones that do are much faster at growing, and so therefore they're the reef building corals, are the ones with the zooxanthellae. Now the algae, because it is a plant, it needs sunlight to be able to carry out photosynthesis. So these particular corals are only found in fairly shallow water, nice and clean, where there's lots of sunlight for them to do that. But yes, there are some other corals, hard corals as well, without zooxanthellae, that live in very deep water and don't need that sunlight. Okay. Then the hard corals are put into the phylum called Cnidaria, and Cnidaria stems from a Greek word neidos, which means stinging cells. So all of the Cnidarians have stinging cells called nematocysts, and there's a picture of one of those on the right hand side there that's actually come out. Cnidaria includes animals such as anemones, corals, jellyfish, and box jellyfish, and blue bottles as well. And I'm just going to stop sharing my screen for a moment so that you can see me. I don't know if you can see me on your screen there. Yes. So the Cnidarians have a body plan, which is a polyp or a medusa. So if you can see my hand, a polyp is basically, it's the animal, it's circular, it has radial symmetry, and the tentacles on a polyp are facing upwards. If the tentacles are pretty short, then, you know, it's commonly a coral. If the tentacles are fairly long, it tends to be anemones. And if you flip it upside down, it becomes what we call a medusa. And that's where you have your jellyfish and box jellyfish and so forth. So they only have one opening. I'm going to go back to sharing the screen again. They only have one opening, which is their mouth and their anus. The stinging cells or the nematocysts are only found on the tentacles. So yes, you can actually pick up a box jellyfish by the bell. I don't suggest that you do that, but all the stinging cells are on the tentacles. And if you have a look at a nematocyst under the microscope, it looks a little bit like an egg, and inside the egg is a coiled up thread, and on the end of the thread is a barb, and inside the barb is some poison. Outside the egg or outside the stinging cell is a trigger hair, and so anything that's brushes past that trigger hair, the barb comes shooting out of the nematocyst very, very quickly and goes into whatever is passing by your finger, let's say. And then whatever happens next depends on what species it is and what poison is within that barb. So for corals, you don't feel anything. For anemones, it's a little bit sticky. For box jellyfish and other jellyfish, it can be very, very painful. So that's what they all have in common for the Cnidarians. Now, the anthozoans is a little bit more specific to corals and anemones in particular. And there's a subclass of hexacorals and octocorals. So if you have a look at the picture on the top right-hand side there, you'll see that this is actually a soft coral. An easy way to know that is to count the number of tentacles that are on each polyp. And so if you have a look at the word octocoral is all the soft corals, and octo meaning the number 8. And so soft corals will always have 8 tentacles or multiples of 8 tentacles per polyp, whereas the hard corals, hexacorals, hexa meaning 6, and so the hard corals will have 6 tentacles. So if you're not sure whether it's a hard coral or a soft coral, I know the daisy coral look a little bit like a soft coral, but they're actually a hard coral. Count the number of tentacles and then you'll be able to see whether it's a hard or a soft coral. Most of the corals only have their tentacles right out at night time. Some, however, do have it during the day. Alrighty, and then our hard corals in particular are called Scleractinia. And then we go to family, genus, and species. So tonight we're going to try and identify at least 6 of the different genus of corals.
I have one question. Are corals nocturnal?
They feed at night more actively using their tentacles. So yes, you could say that they're nocturnal. Yes.
I didn't know that. Wow.
Another way to classify corals is through their growth form. And so here we have, on the left is plate. Another word for plate is laminar. To the right is your boulder coral. It's also called massive, and that doesn't particularly mean that it's massive in size. It just means that it's boulder shaped. Top right is branching corals. Below that's a foliaceous, which forms a whorl. Columnar is sort of branching, but it's only got primary branches, no secondary branches. Down on the bottom there, you have solitary or disc-like free-living corals, much like your mushroom corals. So they're not attached to the bottom, and they're just one single coral polyp. And then to the left, bottom left, is your encrusting corals. So they're the corals that just lay over the top of the contours of the bottom, whatever they may be. Depending on how and where the coral polyps bud, is to what sort of growth form that coral will turn into. So, for example, this is a baby coral, and it starts off as a single coral polyp, and then it starts to bud. So it makes new coral polyps, and depending on where those coral polyps are positioned and how big they are is to how the growth form will actually appear in the end. And just a reminder that some coral species, it might be the same species of coral, and it could be plate, it could be branching. It just depends on the environment in which it lives in, which makes it a little bit hard to classify sometimes. This is a picture of a branching coral, and I've highlighted the corallite, which is the skeletal cup a polyp makes under and around itself. So the corallite, whenever I say corallite, I'm talking about the skeletal structure. If I say the word polyp, it's just the corallite with the living tissue on top. So the polyp is a corallite. The corallite is the skeleton underneath the polyp in which it's made, if that makes sense. So when we're doing taxonomy, we often talk about the corallite as opposed to the living polyp. Here, the polyps have actually spread apart a little bit. So this is a favia coral, also known as a pineapple coral, and I've circled the wall of the corallite. So this is a great photograph for having a look at the different skeletal structures in a coral polyp. So if you just focus on one of those, you'll see that it has a corallite wall, and then inside is some costa and septa over the side, and choanostium is in the space in between the corallites. So you'll hear me using these terms as we go along. So that would have been a massive coral or a bowl of coral. So the first genus we're going to talk about is Acropora or Acropora. I did ask Charlie Perron how to pronounce it, and he said, oh, whatever you want. So you might hear me saying both. I get confused sometimes. The word is derived from the Greek word akron, meaning summit, and it refers to the presence of a corallite at the very tip of each branch of coral. So if you're snorkeling or diving and you see a lot of branching coral, there's a pretty good chance that it's going to be Acropora. But to be sure, just have a look at the very tip of the branch and see if you can see that axial corallite. It'll be right on the very end. And so here, for example, the axial corallite is actually the starting point of every branch. And then as the branch grows up and up, it starts off as a cup, and then it sort of grows into a tube, and then it forms the central axes of the branch. So that corallite at the very end of the branch starts at the very beginning of the branch, and it's like the inside tube to that branch. And then you have these radial corallites, which are all the other corallites that grow around the sides of the branches. And these radial corallites come in a range of shapes, and depending on what the shape is usually what the species is. So we use the shape of the radial corallites to determine down what the species of Acropora it might be. So for example, I've just got a few pictures of different types of Acropora, again. On the left is what it would look like, just a skeletal picture of it. On the right is magnified, so you can really see the shape of those radial corallites. So that's one species. And they just have a really good look at what the radial corallites, what they look like, and then compare them to this one here. So it's still the same genus. It still has the axial corallites and the radial corallites, but those radial corallites are slightly different in appearance, and so it's a different species of Acropora. This is from a fantastic resource by a guy called Wallace, and he came up with all the different shapes categories for Acropora. So that's just you don't need to read all that, but it just gives you an idea of the different sorts of shapes that they're classified into. Here's another one. I think these photographs are just fantastic. And these are all on Corals of the World website that I showed you at the beginning. And this is the last one I'll show you this. And this is the one I just showed you in live form. So this is what you would be looking at. Beautiful colours.
I have a question.
Yes.
Are they in any colour? Or?
Oh, they come in all different colours. Yeah, range of colours.
What determines their colour?
Mainly the algae, mainly the zooxanthellae, determines the colour of corals, which is why Coral Watch uses the colour-coded cards to measure the density of zooxanthellae and the health of the coral. That's how that Coral Watch card actually works. So the algae gives the coral its colour. So when you get coral bleaching, the colour disappears and the coral turns transparent, and what you're actually seeing, the white, is the skeleton underneath. But in this case here with the photo that I've got on the screen now, you'll see that the tips are white, and that's because they're just growing and they haven't yet taken in their zooxanthellae just yet, so they're still white. And there's all sorts of interesting different types. I like this one in particular. And with Acropora, they do come in lots of different shapes. So it's not just branching, although branching is probably the most commonly known one of Acropora, but they do also come in plate and all the ones that you can see on the screen. Is there any questions about Acropora? So really just the thing to remember is those axial corallites and the radial corallites. A different kind of branching coral, which is actually really common as well, is Pocillopora. This is another genus. A well-known common name is cauliflower corals. And these corals do not have the axial or radial corallites like Acropora does, but instead they have very big, what we call verrucae, which are dome-like bumps, and you can see why they're called cauliflower corals. If we have a look at the skeletal structure, you'll see the verrucae there I've outlined. And if you can see the tiny holes on the bumps, they're the corallites. So they make these beautiful patterns. They're very common in shallow areas, particularly in lagoon areas, and they are almost like a weedy species, so they're pretty hardy. If there's been a big disturbance event, they're quite commonly the first branching corals to grab a hold, and they're fairly resilient to bleaching as well. And they come in all sorts of beautiful colours. So you really look at those and you see the bumps with the corallites all over the bumps. The next one is Montipora. So quite often Montipora are your plate corals. They're usually thin, often layered in a leafy sort of appearance and quite bumpy. So either bumpy plate corals or bumpy encrusting corals. There's a pretty good chance it will be a Montipora, and sometimes they're branching as well. So here's just a few examples. You can see the bumpy quite well. This is an interesting one. It's a little bit confused. It's not really sure whether it wants to be a plate or a branching or tubes or what it wants to do. Same with this one. So it really does depend on the environment. To identify them taxonomically, you really got to look close at the skeleton itself. It's certainly not from the growth form, because there's so much crossover between genus.
I do just want to ask a question as well.
Yes.
I personally know a lot of info on coral bleaching. Do you just want to quickly explain, in case anybody in the audience doesn't know
Absolutely.
what the term coral bleaching is? I think it's a little relevant.
Yeah, so coral bleaching is when, so the symbiotic relationship that I mentioned at the beginning between the coral and the plant, the algae or zooxanthellae, if the water gets too warm, or even if it's too cold, or the water quality is not the best, then what happens is that algae starts to create what's called a toxic oxygen compound, and as a result, the coral is like being poisoned by it, and it will throw that algae out. So it kicks it out of its home and the coral's still alive. However, without its zooxanthellae, it loses 90% of its food source. So the algae actually does provide the coral with a lot of food. If the disturbance event goes, for example, if the water isn't warm for too long or it's not too warm, then the algae will actually return to the coral and it will get its colour back and everything will go back to normal. So that's coral bleaching. However, if the water's too warm for too long or it's just too hot and the coral just cooks, then it eventually gets like this fuzzy algae growing over it, and then it dies basically. So yeah, that's coral bleaching. So the bleaching is the coral's not actually turning white; it's losing its color, and so it's transparent, and what you can see is the white skeleton.
Okay, and there's another question from Amanda leading on.
Yes.
How does the coral evict its algae? Are there just tiny boots that kick it out? Or?
Yeah, no, it just, there's some great time-lapse videos where it just slowly starts to lose its color more and more until it goes completely transparent. I don't think they can bolt or anything like that. There might be something chemically happening. Give it the message, but I'm not absolutely sure. Sorry about that.
No, that's okay.
But yeah.
Do you know how they go back to the algae as well? Like, how does zooxanthellae choose which polyp or which coral to come back to?
Okay, so there's actually a whole bunch of different zooxanthellae, and geneticists have put them into clades. So categorize them into clade A to clade I, so A, B, C, D, E, F, G, H, I. And those clades are yet to be turned into genus and species. And each clade of zooxanthellae has its advantages and disadvantages. So a really common clade of zooxanthellae that partners up with the coral is clade C. And clade C is fantastic at feeding the coral, but as soon as it starts to get too warm, it gets the boot. And so there's another clade called clade D, which is slightly different. Clade D is not as good at feeding the coral, but it is more tolerant to those higher temperatures. And so the coral is less likely to bleach. So there's been a few studies where the corals have bleached. They've identified which clade is in the coral initially, and if the zooxanthellae returns to the coral, is it from clade C or is it from clade D? If it's from clade D, then there's a lower probability that coral will bleach in the future. But they do actually change, the different clades of zooxanthellae. Why, we're not really sure. This research is really, really new. We're still learning about it now. When people are breeding corals and putting them into tanks which have zooxanthellae within the tanks, they just enter into the coral polyp. I'm not exactly sure how. It might be a timing thing. Yeah.
Thank you. I didn't even know half of that stuff, so that's really interesting to know.
Yeah, it's a nice, good news story if you have hope that, some corals, they'll bleach the first time and then the second time they may not bleach because they've got that clade D instead.
Nice, thank you.
So here's, there's great diversity in Montipora, some fantastic shapes. Like I said before, you've really got to look at the skeletal structure to be able to identify if it's Montipora or not. And so you can do that, when you're snorkeling and diving. If you just have a look at this is called the coenosteum. So this is the porous skeleton in between the corallites. So the circular bits are the walls of the corallite. Septa are inside, the costae are outside, and then the coenosteum is in between. So the bumpy appearance is usually due to bumps or ridges that grow on the coenosteum between the tiny corallites, and there's lots of different varieties of Montipora. So sometimes the bumps are really small, papillae like this one here, and sometimes they're a little bigger, And you'll notice that the corallites are not on the bumps. So this is not Poecilopora. The corallites are in between. So the bumps are growing up from the coenosteum. They're not, the corallites aren't on the bumps themselves. There could be ridges as well, like this one here. It looks like dripping wax. Beautiful photos. It's a really good website, the Corals of the World. Here's another example. Sometimes there's no bumps at all, and sometimes, like in this instance, there's all sorts of things going on. Okay, how are we going for time? Yeah, we're good. This Parietes is one that you might be familiar with. Is quite well known as helmet coral, or the little micro atolls, or bigger micro atolls, the growing lagoons on the tidal flat. These Parietes corals have really small corallites, less than 2 millimeters in diameter. Therefore, the colonies do appear smooth. And so this is an example of one. They are very slow to grow. So these examples are very old corals here. And you'll see there's all sorts of boring organisms that like to make their homes on the Parietes coral. This is an example of a micro atoll. So basically the coral has started growing up, and then the tide has stopped it from growing up, and so it grows out and around, a little bit like the shape of a coral atoll island, so hence the name. But if you touch these, they are quite smooth when you're feeling them. Lots of things live underneath them and on top of them. Here's a bit of a close-up of the corallites themselves. And there's another one. This is called Big Mama. This is a beautiful big Parietes coral in American Samoa. The typical growth rate of Parietes is about 1 centimetre per year. So you could just imagine how old this particular coral colony is. So these are quite popular for people who want to collect coral core samples, and this is what's happening in this photograph here. So they drill into the coral, and when they take it out, they plug it up with concrete so that no boring organisms can get in, and it's perfectly okay, and it continues to grow. And so they get a big long tube of coral core and then slice it up, and you can see that the coral core has growth rings, much like the growth rings on a tree would have. And so they can age the coral based on those growth rings. And so, for example, in this particular instance, this coral is very, very old. And that's just, you know, if the coral sample piece breaks off, then, you know, it could be even older. But what these are great for is the coral cores operate as a proxy to the climate record, because these particular corals, the Parietes, are fairly resilient to disturbance events such as heat waves, so they're less likely to die. And however, they don't get away unscathed. They usually do bleach, and when that happens, they record that stress event as what we call a high density stress band in the coral core. And so basically you can have a look at your coral core samples, identify where the stress bands are in relation to the years that you identified as the growth rings, and that there is evidence of a stress event for that particular coral. Which is great when we don't have records that stretch way back. This here is a graph of the bleaching susceptibility of 36 genus of Indo-Pacific corals from approximately 68 studies from Kenya to Panama. And I just wanted to put this in here because it shows you that the ones on the left-hand side on the y-axis is the percentage of colonies that bleached. So Acropora and Acropora bleaches quite quickly, as opposed to Parietes. They're a little bit more resilient to bleaching. So that was, Okay. Favorites. I've only got 2 to go, I think. Favorites is commonly known as moon coral. And on the favorites, one of the things that we use to identify corals is we have a look to see if the walls are joined or not. And if the walls are joined together, we call it a serioid colony. And I'll show you an example as to why it's called that common name. So here you can clearly see that the walls are joined, and you may recognize these from when you're diving or snorkeling. They're a very common type of coral. So favorites, you'll have to remember that. And you can see from the close up how those walls are joined together. And this one was one of my favorites. It's just stunning. And the last one is Favia. In brackets it says Dipsastria, which is the genus name if it's from the Indo-Pacific. Recently there was a few changes made to the names of genus and families in coral taxonomy based on genetic findings. And so you may see this one as Favia, of which it's well known as, or Dipsastria, so either or. And common name is pineapple coral. So this is an example of where the walls are separate as opposed to joined, and we call that placoid. And you can see that they're clearly separated, and quite often they're different colours as well.
I have another question, actually.
Yes.
Do all of these species occur throughout the world? Are there different tropical, subtropical, or Arctic species? Is there such thing as Arctic coral?
There is, yes. So these photos are from corals of the world. So these are found in various places. And if you go on the website and have a look at the genus, it has a bit of a distribution map there as well, which shows you where they're commonly found, or where they're found and where they're not found. So these, I don't know these particular species, because each species is different. So you'd have to look at each species and see where it's found around the world.
Okay.
But these are I picked these because these are probably the most commonly found ones. Certainly here, but yeah, it could be anywhere around the world, depending on the species.
Nice, thank you. How many different types of species of coral are there?
Oh my, lots. Lots. Yeah, lots. Thousands and thousands. Yeah.
Oh wow.
Yes. And so here's just a picture showing the difference between the two, with favorites with walls that are joined and Favia with them separate. Okay, I did say at the start that I was going to show you this slide again and see whether or not you can identify them to genus level. I've given you a couple of hints on there to see if that rejogs your memory. Even if you don't remember the genus name, at least you'll be able to know what to look for the next time that you go snorkeling or diving.
I have also had a lot of people asking, just because this presentation is fantastic, will you be able to make this publicly available?
So you're recording it, yeah?
Yeah, I have told everyone that it will
Okay.
be recorded and put on the YouTube, and I've linked your
On
website
YouTube.
in as well.
Okay, perfect. Yeah, and I'll probably put it on my website as well.
Okay, cool.
For the students, yeah.
Well, I'll repost the website link, and for anybody that wants the presentation, it will be on there.
Yes, I'll put the PowerPoint on my website under the YouTube link.
Thank you.
That's all right. All righty, so hopefully you've had time to see if you're right or wrong, and there are the answers. So Acropora, you've got the axial and radial corallites. That's the most common branching coral. Another branching coral is your Poxlopora. where you have the verrucate bumps and the little tiny corallites all over those bumps. Montipora has a range of growth forms, but well known mostly as being the bumpy plate or bumpy encrusting coral. Porites is your very slow growing, very dense skeleton with the very tiny corallites, less than 2 millimeters in diameter. Favorites is your corallites with the walls that are joined. And favia, the walls are not joined. Okay, I do have a video here, but I might just do it this way. Can you still see my screen?
Yep.
Yep.
That's coming up perfect.
All right. Hopefully no ads pop up in the process. Is that full screen yet? There we go.
Yes.
Hopefully you can hear the music. It's very short, but this is some time-lapse video of a cropera. Enjoy.
And would these photos be taken over days, months, years?
So you see the tentacles going in and out, so that's day and night.
Ah, okay.
They grow fast.
They do. Oh my Lord. Wow.
Great. And I think that is it. I did have more genus, but I don't think we've got time for more. Yeah. Thank you.
No, I want to thank you.
Oh, hang on. Sorry.
Are you still here?
Yep, I'm just shutting that.
No, that's okay. You've stopped sharing screen anyway.
Okay.
I want to give a big thank you once again for presenting. This was extremely informative, and there are lots of comments there saying thank you as well. Attached is Gail's website, marineeducation.com.au, and I'm just about to post another link as well. If anybody's bored and they want to hop on to a Google Street View of underwater, seeing one of those photos made me remember awesome website, yeah, that I found of these awesome cool corals. It's like walking along a street in a different country. You can go scuba diving from the comfort of your own home, really. There are heaps and heaps of different dive sites as well that you can choose from all across the world if you do some research, and there are heaps of other websites like the XL Caitlin Seaview Survey. If you Google them, they give you 360-degree photos and videos of these cool dive sites. But there's heaps in Australia, in the Caribbean, in the Coral Triangle. If you like coral, you don't even have to go on the water anymore. Otherwise, I hope everybody enjoyed this very interesting talk from Gail. Thank you very much for turning up, everyone. It's been awesome. Please remember to like us on Facebook and keep up to date. We will be having another talk next month about sponges next time. So it's very close. And I hope everybody has a great night. Thank you.
Thank you, Pablo.
Thank you.
Worries.
Thank you, Gail.
My pleasure.